So I'd like to welcome everyone to this month's ReproNim webinar. We are super delighted to have a presentation by Mike Yassa and a follow-up commentary by our own David Keeter on the topic of early life adversity and brain development. Mike is an associate dean in the Office of Diversity and Equity and Inclusion in the School of Biological Sciences at UC Irvine. He's a professor in many departments, including neurobiology and behavior, neurology, psychiatry and human behavior, and the Department of Psychological Sciences. He directs the Center for Neurobiology of Learning and Memory, and he's the director of the UCI Brain Initiative. They do all sorts of work in various imaging modalities, structure, functional, diffusion MRI, PET, EEG, intracranial recordings, and exploring the brain architecture at very many different levels of detail. He has his BA in neuroscience from Johns Hopkins, a master's in psychological and brain sciences from Hopkins, and a PhD in neurobiology and behavior from UC Irvine. And without further ado, I will turn the floor over to Mike. Thank you for joining us.
Thank you, David. Thank you for the invitation, and I'm delighted to be here today and share with you some of our work in the Conti Center. As you've already heard from David, David Keeter, works with us at the Conti Center. So some of these things are subject to many discussions and debates and conversations, but I'll share with you kind of a few highlights of what we've been doing with imaging data at the center, and maybe some challenges and some issues that I'd love to have some discussion and some insights from the group on. So just to give you a little bit of background, we have a NIMH-funded Conti Center. I'll tell you a little bit more about it in a second, but our imaging core team, as you can see here on the left, includes Dave Keeter, Andy Obenhaus, Jared Rasmussen, and then a brilliant MD PhD student, Bianca Leonard, and then Mithra Satish Kumar, who's our imaging analyst and database administrator for our imaging data. A number of alumni have also contributed to this work, most notably Sarah Kark. I'll share some of her work today, and Steve Granger, I'll share some of his work, and then Robert Gersari. Those are the three sort of big highlights that I'll share today. So what is the Conti Center attempting to accomplish? The overall goal is to look at brain development with an eye out for early life experience. And the idea is that early life experience poses either vulnerability or resilience onto the developing brain, according, of course, to the nature of those early life experiences. And in particular, we're focused on parental influence and environmental signals, and the degree to which their unpredictability, so in other words, a chaotic early environment, can predict aberrant maturation of brain circuitry. And we have several brain circuits of interest, ones, of course, that are involved in sort of higher order cognition, memory, and decision-making, but also circuits that are related to processing of emotional information and reward information. So I'll show you some of the highlights from this work today. Our imaging core is quite versatile, and I think it is a little bit different from a number of, so the way that we envision a core, usually it's that it's really focused on service and provides access to databases of imaging and metrics and so on to allow for investigations to be done really by others outside of that core. But we tend to serve a dual purpose. So we are certainly tasked with establishing this very rich data set of whole brain imaging metrics, using a variety of different approaches, and then working with our colleagues in other cores to test hypotheses. But we are also very interested in testing hypotheses within the imaging core. So we tend to be a hub where the neurobiological hypotheses are being tested. And most of these hypotheses are related to patterns and trajectories of brain development as they are impacted by early life experience and early life adversity. And we know that there's sex-differentiated patterns in brain development in general, but they tend to be also amplified in the context of early life experience. So that's a big part of our focus. So let me share with you some key discoveries and highlights. So I'll share three very short stories today. Two of them are, one is published, one is in prep, just about to get submitted next week, actually. And then the other one is partially published. So we have one paper out and then another one that's in prep. Starting with the first one that is really focused on the use of diffusion imaging data. So the title here is Unpredictable Sensory Signals Are Associated with an Imbalance in Cortical Limbic Circuits, which is linked to memory performance. This is the work of my former graduate student, Steve Granger, who has now gone on to postdoc at Harvard McLean. And the work that he did was really focused on understanding both the role and contribution of the unsignificulus, but also understanding how this particular pathway in the brain might develop in an aberrant way in the context of early life experience. So if you can see my cursor pointing here, the unsignificulus is this pathway that somewhat interestingly kind of follows the curve of the medial temporal lobe and up into the frontal cortex. It's a pathway that we believe really only exists, at least to this extent, in primates. So we don't have a rodent analog really of the unsignificulus. But there are other pathways certainly that do link the temporal lobes to the frontal lobes, in particular the cingulum, which we'll talk about a little bit. So this pathway is involved in memory processes, and in particular, the intersection of memory systems and decision-making systems. So it's really how do you make use of memory and update the use of mnemonic representations. And the question that we asked is, how do unpredictable sensory signals during infancy affect the integrity of the unsignificulus? And without going into too much detail about how unpredictable sensory signals are measured, that's a whole other talk. And I can't take any of the credit for that. This was really the ideas and development of my colleagues at the Conti Center, led by Tali Barham. And it's really focused on the degree to which sensory experiences may be predictable or systematic, or have an element of chaos and randomness to them. And there's a very nice mathematical way to measure that using Shannon's entropy that we're not going to go into too much detail about, but it is sort of measured in a quantitative sense. And our approach here was to assess structural connectivity of the unsignificulus, and as a control, another pathway that connects the medial temporal lobe and the frontal lobe, the cingulum, using diffusion imaging. So this study involved 73 mother-offspring dyads, 30 girls and 43 boys. And the reason we use dyads is because we're looking at maternal sensory signals. So even though the imaging that we're looking at is all done in the children, the assessment of early life experience is based on the interactions between mother and child. And the unpredictability in these maternal sensory signals was assessed at 6 and 12 months of age, but the MRI scans that we're looking at are about 10 years later than that. Children were also administered something called the mnemonic similarity test, or MST. This is a test of hippocampal pattern separation. It's a very robust memory test. And in particular, it's aimed to examine how well we can do at differentiating very similar memory experiences to have accurate recognition. So it's a very challenging version of a memory recognition test. And we used this was a multi-B shell acquisition. We used what's called QSDR, or Q-spin diffeomorphic registration, in DSI Studio to do sort of a model-free version of tracking based on fiber integrity. So here's the protocol, or the method that Steve developed to be able to use anatomical landmarks to constrain the charactography within anatomical regions of interest, to really limit it to just these pathways going from the temporal lobe to the frontal cortex. He had exclusion zones at the back of this pathway and at the superior angle of this pathway, also to ensure that it only included those fibers that are really kind of anatomically consistent with what we believe the unsignificulus looks like. And then within that pathway, he assessed GFA, generalized fractional anisotropy. And here you can look at this on the left. You can see that there's a relationship that is maybe a little bit steeper, a little bit more amplified in girls than in boys, with unpredictable maternal sensory signals. So the higher that score is on the x-axis, the more unpredictability in their sensory environments there was. And you can see that tracks with a higher GFA, so a sort of a higher integrity in the unsignificulus. When we looked at the cingulum, hippocampal cingulum, as essentially a quote control pathway, a pathway that we did not suspect would change in the same way, we don't see this relationship. But then Steve also did something really interesting, which is look at the ratio of GFA to cingulum. The idea is that the frontal cortex is receiving input from the medial temporal lobe through these two pathways, and maybe it's the balance across the information that is passed on through these pathways that is really consequential. So he calculated the ratio of the unsignificulus GFA to the hippocampus cingulum GFA. That's what you're seeing here on the y-axis. And that was also positively correlated with unpredictable maternal sensory signals. So it suggested to us that it's actually an imbalance in this particular circuitry that may be associated with early unpredictability. Now, the question is, well, so what? Does it actually have any consequence for cognition, for emotional development? And we were able to look at one aspect of cognition here. This is the memory test that I mentioned earlier, the mnemonic similarity test. And you can see here that during encoding, you're looking at pictures of everyday items. During retrieval, you're seeing some of the same items. Those are the targets. Some brand new items. Those are the foils. And then some lures. These are challenging, similar lures that are similar but not identical to items they've seen before. Like this coffee mug is not exactly this one. This glove is not exactly this one. And the question is, do you do well at saying this item is similar but not identical to the one you've seen before? And we quantify that using the lure discrimination index, which is essentially an index of how well you're able to discriminate those lures. And you can see that that's inversely correlated with this ratio. So in other words, the higher this ratio is, which we've already established is associated with higher levels of sensory signal unpredictability, is also associated with lower performance on lure discrimination. And here you see a mediation model that links unpredictability to lure discrimination. And there's a significant mediation going through this pathway, which is the ratio of unsignificulus to cingulum GFA. So all in all, just to summarize this story, unsignificulus integrity about age 10 is associated with unpredictable maternity sensory signals during infancy, about 6 to 12 months of age. This effect appears stronger in girls, although that difference is not at this point statistically significant, but we suspect that it's real because we've seen it in many other analyses that we've done as well. This indicates a possible circuit imbalance with the cingulum. So corticolimbic pathways have to kind of constantly be balanced in terms of information flow to the frontal cortex. And we suspect that this circuit imbalance with the unsignificulus being exaggerated in terms of its contributions to frontal computations may be a culprit here. And it appears to mediate the relationship between unpredictability and poorer performance on a memory discrimination task. Of course, natural next avenues to try to investigate would look at the development of those children into early adolescence and whether this is also going to predict maybe the onset of some psychopathology. But those are some of the things that we're sort of planning on doing in the near future. Okay, let me move on to the second story. So here we're looking at a slightly different version of unpredictability. Here we're looking at another kind of entropy that has to do with mood, and in particular, again, maternal mood. And we're talking really about the degree to which assessments of maternal mood during the gestational period are different from each other, or are in some ways a lot more random than systematic. And we chose to look at the salience network in particular because it is one that is really important for socio-emotional regulation. And past work has also linked early life adversity, not this particular type of adversity in terms of unpredictability, but other types, experiences of abuse and neglect and so on, has seen that the salience network seems to be implicated in that. So we asked the question, how does maternal mood dysregulation during gestation affect the integrity and flexibility of salience network functional connectivity? So here we're shifting over to look at functional data rather than diffusion imaging data. And our approach was capitalizing on data that had been collected in the Conti Center a number of years prior. So the idea was, let's use however much data that we have available to us to really assess functional connectivity. We had a sequence of 3 scans that were administered to children at that age. There were 2 resting state scans flanking an emotional memory task. So in other words, one approach could be, well, you could take a look at the 2 functional connectivity or resting state functional scans, average them together and get beautiful measures of connectivity. And that's certainly something that we were able to do. But having this emotional memory challenge in between gave us an opportunity to be able to look at dynamic flexibility of these networks. So how much of a change do you get over time with this emotional challenge? Do you get sort of an upregulation in some networks and then a downregulation again after the task is completed? So it allowed us to look at dynamics across the 3 scans, but we also wanted an independent measure of dynamics even within the scan. So we used ALFFs to be able to do that. And I'll show you data from both of these approaches. So here we're looking at 138 mother-child dyads. Mean age here is 13. And as I mentioned earlier, the mothers completed mood assessments, 5 of them during the gestational period. And we really looked at mood entropy by computing Shannon's entropy to the distribution of responses in each of these mood questionnaires. So it's a very, very different kind of early life experience because it is really kind of during the gestational period. And the idea is that this dysregulation of maternal mood during the gestational period might have impact on the development of the fetal brain. The children underwent fMRI scanning during early adolescence, so it was really about the age of 12 or 13. And you can see here the experimental design. There was the resting state scan 1, then the emotional memory challenge. So this was a challenge where they're just looking at either fearful faces or neutral faces. And then the second resting state scan. Activated regions during the viewing of fearful and neutral faces were sort of the usual suspects. You see the amygdala, you see the cingulum, lingual gyrus, all of the regions that we typically kind of see that are involved in a salience processing kind of network. Then this is the work by Robert Gersari, I should say, who was working in my group up until about 2 years ago, and then he started graduate school at WashU St. Louis. He's working with Deanna Barch now. And he pulled out basically all of the different networks using an ICA approach. So we have all of the networks here, the typical kinds of things that you see, a default mode network, salience network, visual network, and so on. And looking at the salience network in particular, he was able to associate that with prenatal mood entropy. And again, this is a significant relationship, although there's a lot of variability. Now here, he actually looked at all 3 scans for every participant. So you can see here, every one of these little spaghetti lines is essentially the 3 scans, one sort of stacked on top of the other. And what you immediately observe is that some are much more variable than others, which is really interesting. So that's something that we want to kind of take into consideration as well. So what he was able to do with the help of Anton Palma, who is a statistician working in our group, is cluster people into 2 clusters using a latent class mixture model. And you can see that there's one cluster that is highly invariable across time. So this would be the level of integrity of the salience network here. And this is just the overall level of connectivity. And you can see that there's not much of a change over time from REST 1 to amygdala to REST 2. Whereas there was another cluster that was pulled out with this approach that is highly variable. So you see a lot of dynamics that are ongoing, either upregulation or in some cases a downregulation. This was also correlated for prenatal mood level and sex and age and so on. So you can see here the things that kind of popped up were the integrity of the salience network. That was very different across 2 clusters. Clearly, prenatal mood entropy and mood level, those were also different. Those are the predictors, and we'll talk about that in a second. But also the ALFF measures across those 2 clusters of individuals were different. So in other words, the variable cluster across scans was also variable within scan if you look at these low frequency fluctuations. Here's another way to be able to visualize that with ALFFs. So you can clearly see here that this cluster, if you look at just the variability or the dynamics within scan, is a much, much higher level of variability than within scan variability for the invariable cluster. So both of those measures kind of gave us a similar story, that in general, the degree to which this network is dynamic and sort of shifting its connectivity level is dependent on which cluster you belong to as an individual, and that was related to prenatal mood entropy. So to summarize this, salience network integrity in late childhood, early adolescence was associated with maternal mood dysregulation. Here, dysregulation is a catch-all to indicate that it wasn't just maternal mood entropy, but also maternal mood level. So that captures in it depressive symptoms and so on for the mothers. But those 2, I should say, even though they are correlated, they do contribute independently to the variability. So there's a kind of an independent contribution to entropy above and beyond what we see with overall mood level. This latent class mixture modeling allowed us to take out sort of two distinct clusters here. The invariable cluster seems to be the one that was exposed to higher levels of prenatal mood entropy and overall negative mood. And it was also considered rigid using quantifications of ALFFs, or amplitude of low frequency fluctuations, that capture the within scan dynamics rather than across scan dynamics. And these relationships withstood having sex or age as a covariate. So this was one of the actually rare analyses in which we didn't really find much of a sex differentiated effect. Okay, now let me go on to the third piece, which I think is really, really exciting because we've started to kind of look at new anatomy here. So as many of you know, the paraventricular nucleus of the thalamus is one of those regions that you've probably been seeing a little bit more in the literature these days. It's gaining a lot of attention. We think that it's a potential integrator of emotionally salient early life experiences. And I say salient here to be a little bit more inclusive. It's not just adverse experience. It could also be rewarding or positive experiences, although that work, there's not as much work in that particular area. What you're seeing here is an illustration of the connectivity. This is anterior and posterior PVT, and connected to a variety of different regions, including the nucleus accumbens, including the bed nucleus of the stria terminalis, the central amygdala, certainly frontal lobe, infralimbic and prelimbic cortex. So those kinds of influences are both getting input from PVT and also are having their own input pathways lead to PVT. So it is this hub that is able to synthesize and maybe process that kind of information. And one of the things that we try to do is, the PVT has never really been looked at in humans before. It's a very, very small structure. So Sarah Kark, who was a postdoc in my lab also until about a year ago, she moved on to a very, very nice position in industry. So she's continuing to do some really fun things, but maybe a little bit less of the imaging and neuroscience bits and more on biomarker science. But what she did in the lab was really just transformative. She thought she's going to take this on as a challenge. She looked at the Human Connectome Project data, 7T and 3T data, and really generated the first evaluation of functional connectivity of the human PVT. And you can see this paper did get a lot of views and a lot of citations already, even though it's only been around a short time. But it really was path-breaking because she provided all of the methods and approaches. So all the data, all the code, all the masks that she generated are all available online on Open Science Framework. So you're free to take a look at that. But here's in essence what she set out to do. So she took 121 adults, 22 to 35 from HCP, both 7T and 3T data. So this is a subsample that has both 7T and 3T data. And she used the 3D atlas of the human thalamus that was published not too long ago, based on just the postmortem anatomy. And she kind of created masks based on this atlas and was able to quantify PVT resting state functional connectivity in a way that controlled for the immediately surrounding other nuclei that are associated with the thalamus. And I'll show you exactly how she was able to parse those apart in the next slide. But you can see here, here's the overall thalamus in the very bright fuchsia color, and then the PVT itself is this tiny little yellow piece. So it's only really a few voxels across this extent, not a huge structure by any means. So identifying it and trying to understand whether its connectivity is different from other thalamic nuclei was a particularly interesting challenge. But she developed a really nice method to do that. And what we can tell based on these functional connectivity studies is very similar to what we've known from the anatomy, is that the human PVT is functionally connected to the amygdala, the hippocampus, the accumbens, the VTA, and then hypothalamic regions, brainstem regions, which is very much consistent also with animal models and looking at this particular anatomy in rodents. Here you can see that one of the largest connectivity patterns that she observed was really with the default mode network. So the PVT has its highest level of connectivity with DMN, and that's what you're seeing here in the polar plots. The nice thing about this analysis is that if you start to look at other parts of the other parts of the thalamus, for example, other nuclei, CEM, CL, and PF, parafascicular, all of these nuclei that are part of the thalamus, you actually see very different connectivity patterns. So even though they are in such a close locale, she was able to extract out a different functional connectivity pattern that you're seeing here. So, just as an example here, the parafascicular nucleus is much more interconnected with the ventral attention network than it is with DMN, for example. There's virtually nil in terms of connectivity with DMN. So we do think this is real, given that the pattern is so different across the different nuclei. And then the question is, well, can we do the same thing at 3T and at least get elements of the same findings? And that's what she embarked on next. Here you can see both 7T, 3T, and then the overlap. But even at lower resolution, 2mm resolution, one can obtain PPT connectivity reliably. And we do see patterns very similar. Again, connectivity with medial prefrontal cortex, precuneus, amygdala, hippocampus, VTA, accumbens, and hypothalamus. So it was a beautiful replication, obviously in the same people, but here you're looking at 3T data, which I think makes this kind of enabling. So then we started to apply this. So then as she left the lab and went on to greener pastures, then I had a MD PhD student in the lab take this on, Bianca Leonard. So you're seeing some of her work here, trying to apply this same method and approach to our Conti Center data. So here she's looking at 178 children, 91 boys, 87 girls. I shouldn't say children, I should really say mostly adolescents, 9 to 17. So it's a broad age range. Here, the image resolution is not as good as HCP. It's a 3mm isotropic resolution. The idea is, can we see some of the same elements of the things that Sarah was able to observe in the HCP data? And the answer is yes, to an extent, we do see that. So here we see the Kark et al. Paper, very robust connectivity in DMN. And an element of that here in this sample, obviously not as robust, not as large in terms of clusters, but the spatial localization is actually quite similar. So you do get DMN, you get a lot of the frontal regions that are interconnected. So as a sanity check, that made a lot of sense. Same pattern, but it is a little bit weaker. Of course, this was the adult brain, and we're looking at children and adolescents here. One of our next big directions is to look at HCPD, the developmental sample, so we can try to have a comparison also in the developing brain to know whether this is truly a developmental phenomenon or just related to the quality and resolution of the data itself. But one of the interesting things that she was able to do was look at patterns with age and sex. So here you're looking at patterns with age, so positive change with age, negative change with age. And one of the things that you see with age is that there's sort of a shift from the sensory regions that are a little bit more in the blue here to frontal regions that are a little bit more in red. And when you look at a sex difference between boys and girls, you do see females are higher in terms of their overall level of PBT connectivity with different regions of not just the default mode network, really overall across all connectivities. So it seems to be the case, and a lot more of the frontal connectivity as well. It seems to be the case that this particular pattern is a little bit more well-developed in girls than it is in boys, at least at this particular age range. And that might have to do just with developmental age and the way that adolescence is really kind of at different stages in boys and girls. This is a slightly more granular view, but I'll just go ahead and skip that. I mean, again, you see more females connectivity than the males, but all of the data is kind of being looked at now with an eye out for is there an effective motion and all of these other things to make sure that it's fully vetted. One of the things that was really interesting is we've looked at this idea of dynamic connectivity before. I see a question, a hand up, Tafhim. Yeah. I'm sorry, I'm not hearing you.
Maybe we should go forward and we'll come back to questions later on. Oops, she's unmuted. We can't hear her.
Okay. Okay, let me go on and then I'll come back to questions just a moment, like David said. So one of the things that we wanted to look at again is, we understand that there's going to be dynamics that are different from in terms of resting state scans, but we do have longitudinal data on these children, and we want to understand the developmental trajectory. So does it make sense for us to just use the cross-sectional data that we've looked at and look at it at different ages, or should we look within individual across scans? And of course, the answer is longitudinal analyses should always be more powerful. But we ran into an issue, which I'd love to discuss with the group here, is that longitudinal resting scan fMRI is just, it's a mess. There was just too much variability to deal with across scans. And you can clearly see here, if you differentiate, male versus female, both of them are highly variable. This is looking at PBT with hippocampus, PBT with amygdala. So those are areas in which we saw differences when we looked at things cross-sectionally, but when we looked longitudinally, there was just too much variability. And it's not clear whether this is truly, developmental differences, maybe that's unlikely, versus just resting state scan variability within individual. There wasn't a clear path, there wasn't a clear trajectory over time, which was a little bit of a disappointment. But it kind of brought up the question of, when we do longitudinal analyses with structural data, one of the things that we are very sensitive to early on in the days of doing that is that you can't just use the cross-sectional approaches. You have to develop a longitudinal pipeline that takes all of the different time points and average them into a single subject template, and then you kind of propagate out all of the different time points from that template. So you avoid that first time point bias. That's a very, very known bias in structural imaging. But I wondered if maybe some of the same approaches can be propagated into resting state fMRI. And I haven't had time to kind of sit down and think about exactly how to devise a method to do this. And I'm unsure whether or not that would resolve the problem. But again, that's an issue that we've struggled with in terms of, whether these data are reproducible across time within the same individual, and whether we can make sense of longitudinal trajectories that way. So just to summarize the PBT analyses, from the work that I showed you from both Sarah and Bianca, it's clear that we can resolve functional connectivity and anatomy in human MRI at 7T and 3T. The connectivity is largely similar to what we see in the rodent, which I think is a very good thing. In the adolescent brain, the connectivity patterns that we see are present, but they are weaker. We'll really be able to fully assess that once we start to look at HCPD data, which is happening right now, and be able to tell whether some of that is influenced by the lower resolution that we have in these data. We do see that with respect to age, there's an age-related shift in PBT connectivity from more sensory regions to more frontal regions, and there's stronger overall connectivity in the girls versus the boys. So this pattern appears to arise earlier in girls than boys, so it suggests that there's sort of an earlier maturation, or earlier frontal maturation at least. But with respect to longitudinal functional connectivity data, I'm still kind of at a loss a little bit. How do we enhance the reproducibility of these approaches? Is there something that can be done to try to kind of get them in the same temporal space? Is there a signature from individual to individual that doesn't really change over time for the resting state connectivity? Those are all things that we're sort of grappling with right now. So with that, I just want to thank the direct contributors to this work. Obviously, there's lots and lots of collaborators you're seeing here on the right, but the work that I showed today was directly driven by Bianca Leonard, Sarah Kark, Robert Jassari, and Steve Granger. So with that, I'm happy to open it up for discussion and questions. And Dave, feel free to obviously chime in as well.
Yeah, so I'll make sure that if Dave has comments to make here, and you're go for it, Dave.
Well, I'll wait for questions about the science first, and then maybe I can speak a little bit to, how some of this work gets done within the lab in terms of the informatics, some of the tools that ReproNim has used, and then maybe Mike might want to comment on some of the difficulties of using some data from publicly available data sets, in this case HPC.
Oh, I'd be happy to do
or
that.
HCP, sorry.
Yeah.
Not the cluster.
Yeah, that's right. That's right. I mean, I can say something to that. I won't take too long and belabor the point. What I have to say is quite pithy. It's just, there's a challenge in terms of being able to use public repositories, and in particular HCP, in a format that allows us to do the analyses that we really needed to do. And we wanted things to be in as raw a format as possible, and to be in nifty format, so we can apply some of our not just the Khan toolbox kinds of things that we use, but our in-house scripts. And we got a lot of resistance from the HCP team in terms of providing data in that format. They were sort of vehemently opposed to it in principle and suggested that we really need to use the surface-based file format rather than the Nifty format, which was a little bit of a disappointment. And then they also insisted that doing our own pre-processing is never going to be as good as the pre-processing that they have done. But then they didn't really share the details of what they have done all that well. So it was more like, no, we give you the data set that you should be using. You should not be using the raw data and doing anything with it, because we've had some challenges. And while I appreciate that, and obviously there's great reason to not put anything out there that is going to mislead the community. This is not our first foray into imaging. We've been doing imaging for a long time. I think we're capable of looking at raw data and assessing issues with it, and then having our own pipelines deal with it effectively. If nothing else, we could do a comparison and see which ones do better. So that was a little bit of a disappointment. We ended up circumventing that just because Robert had direct access to the data set, so we were able to get it anyway. But it would have been nice to kind of be able to work with the HCP team to get things in the format that we needed and not have that be dictated, for lack of a better term. And I know we're all colleagues here, so hopefully I'm not offending anybody here, but that was, I told my colleagues over at HCP my feelings about that as well.
Well, I think that's a nice example. I mean, it speaks to the availability of data set, open source data sets in the raw formats, along with various levels of pre-processing. Yeah, I mean, I would agree with that perspective as well. I don't know what others might think on the call, but I think for making data sets publicly available, and it's for the sake of open science, we all would like to try potentially different processing pipelines and our own tweaks on those pipelines. And so making the raw data available in a well marked up, well annotated way is super critical, I think, for the advancement of science. I don't know, Dave, if you have any, or Kennedy, Dave, if you have any additional comments on that.
Well, only sort of echo sort of what I think you've both already said. There's two different things that are often going on. One of which is often people will take data and do the exact same first pre-processing. Again, so many people take data and do fMRI prep or do MRI QC or do FreeSurfer and things like that. And there's a huge value to doing that once and having people start from that for all the things that they want to do that. That being said, any one pre-processing pathway you take is not the end all and be all. And again, like you're arguing, the ability to do that pre-processing bit different ways and explore that source of variability is equally important. So again, we sort of have to have it both ways, ultimately. But again, these big resources, are even the big resources are underpowered, in terms of their person power and trying to get as much out that is usable by the end community, and that is important to them. But again, so it's a balance and it's a challenge, and because we have to be patient and insistent at the same time to try to get to these things. Can you say again how you got circumvented this problem? I heard what you said, but I didn't quite catch it.
I'm trying not to throw anybody under the bus here, but my former student who was part of the HCP team, so he had kind of insider access to it and had all the permissions in place and everything to be able to access it. So when I was lamenting the issues that we had with this, when he said, well, I can certainly just take your pipeline and apply it directly to raw HCP data and provide you with that process data. So we just went ahead with that, and now we have it, and we're going to start analyzing it very soon. So it was a little bit, it would have been nice to kind of have that data in the right raw format available for download. It's not right now. And it's not even just compatibility with our in-house things. So I can imagine okay, well, they can't quite cater to everybody, but we were talking about using, the Con toolbox, which is a very, very standard kind of thing. And it wasn't in a format that the Con toolbox can handle. And the response was, well, maybe you should use something other than the Con toolbox. It's like, well, we kind of like the output from Con toolbox. It does a lot of things for us, and we've built a lot of scripts on top of it, right? So instead of just adjusting our entire pre-processing pipeline, all the scripts and all the training that our students have gone through, to adopt a new process, it would have been a lot easier to just have data in the right format. And even if it wasn't, like I would have been okay with minimally processed data because I trust that, yes, they've taken care of a lot of the issues, whether it's, distortions or motion correction or whatever, but then provide the minimally processed data in a couple of different formats. To say that the Nifty format, which is I believe still the standard, at least in many circles, is not the right format that you want to provide things in because you want to move the whole field to a different format that you like to use is a little bit, I think, maybe not justified.
Yep. Can I throw a quick question at the longitudinal analysis? Can you quickly, I may have missed it, how many scans and what are the ages of the scans in your longitudinal components?
Yeah, so let me go back to that. So this analysis here had, 178. So 178 children had up to 3 scans, right? So some have 2, some have 3. So those are the spaghettis that you're looking at here.
And those, if they had 3 scans, that was a month or a week or a year apart, or what kind of timing?
No, it was definitely a couple of years apart. So they weren't like in close succession. But interestingly, even if they were in close succession, I want to show you an analysis that Robert did some time ago. This was one of my extra slides here. So he looked at resting state scan variability within participants. Just being able to say, hey, we have run 1 and run 2 in the same session. How variable are those two? And you can see here that there was a lot of variability. Here's one subject in particular that had an R value of 0.16, right? So not very correlated. And here you can see one that is much more correlated within participant. And he kind of evaluated that just in terms of Pearson correlation. The mean was really kind of a moderate correlation, about 0.47. So even within, now one can say, look, that's probably driven by the fact that you've got an intervening emotional memory challenge or emotional challenge or whatever it is. It's possible. But if even within the same scan session, the correlation across 2 functional, or resting state scans is only about 0.47 on average. I don't know that we can expect better than that in longitudinal analyses.
Right.
Now, granted, this may have to do with this particular data set and movement and the fact that they're children and all those things. But I'm interested to see what the group thinks in the grander scheme. Should we be looking to resolve some of these challenges with resting state data over time?
Well, the short answer is yes, of course. But how is the challenge? And this actually gets a little bit to the sort of the question I was going to ask. But going back to your spaghetti plots, those particular spaghetti plots, do those come from the pre-emotion or post-emotion? Were those spaghetti plots from the same?
These ones here.
Yeah, from the same.
Yeah. So for this particular one, they ended up taking run 1 and run 2, the 2 functional scans within that same session, averaging them together.
Okay, and these are specifically for the PBT.
Yes. So on the left here, you see PBT connectivity pattern with the hippocampus, and here you see it with the amygdala.
And so I guess the thing I was interested here, because of the PBT being a particularly small, thing, I was curious if the spaghetti plots are quite as chaotic with something big, a genu of the corpus callosum. I don't know, something that might be a little bit spatially more robust too, since the size of this, may be an issue. I sort of think
that's a very fair question. That's a very.
I think your spare slide kind of answered though that it's quite variable when you look at every pair of connectivity.
I mean, honestly, so I kind of want Dave to speak to this as well, Dave Keeter to speak to this as well. But I feel like in this sample in particular, we have not had much luck looking at longitudinal imaging and being able to really extract trajectories at all. And the same is true for structural data, I believe. Dave?
Yeah, I mean, they're certainly not consistent across individuals. There may be some trends like, even we're seeing here, but you end up with spaghetti plots. I mean, you don't end up with a consistent longitudinal change that one would expect. And so it's curious as why that is. I mean, there's lots of possible explanations here. Aging, time between scans, development, and so forth. right down to scanner artifacts, as Mike suggested. But yeah, it's definitely a challenge.
And one thing that, initially when we thought about this, we thought, okay, we have some that have, say, 3 time points. So we can look at whether or not there's really kind of a trajectory across those 3 time points. And maybe some will be flat, some will be trending, but you're not going to do this, right? Like that's not really biologically plausible to kind of go up and down, or to, there's not a lot of developmental events that would lead to that. So when you see that kind of pattern, the up and down, that tells you, hey, maybe there's some variability in the data here that's not related to development. And I think we saw a lot of that with the structural data, and certainly we're seeing it with the functional data.
No, yes, very challenging question. I mean, may not solve today, but it's good for us to be working on.
I know, I didn't expect to solve the problem today.
Oh, good.
But I just kind of wanted to put on your radar. It's one of those things as we're thinking about longitudinal data in general, like how do we evaluate the role of biology versus just the reproducibility of imaging data?
Yep. Other questions from the audience? And yep, go ahead, Dave.
Oh, I was going to say, if there aren't any questions, I could say just like 2 minutes about the Conti Center and a little bit about what we've done in the past and currently doing with the data, if that's of interest, and if not, that's fine too.
My share here that way.
Yeah, why don't you say a little bit about that?
So, I mean, I can even share a slide or two if you want, but if not, it's fine. But so there's been 2 Conti centers, the renewal and the original center, and some of the data Mike showed today was from the first Conti Center 1.0 is what we call it, right? And so in that setup, that was sort of started before bids, so we didn't really have bids, but what we did was we during the grant term of that center, bids came online. So we moved data into bids and did the regular, what seems to be a regular ReproNim thing now, where we annotated the bids data and then we converted it to graphs with an IDM. We did that for the human neuroimaging data, and we also did that for the rodent neuroimaging data. And we use those graphs, and we used a graph-based database, much like the Repro Lake is right now. And I think it was an early Repro Lake. And we put all that data into the Repro Lake, and then we started asking questions of that data. And we did some transformations on the graphs in the background that said, well, age 13 of a adolescent is roughly equivalent using this transform to this age in the rodent. And I'm not going to say what P age that is because I have no idea. But we built that in the background, and then we had this nice little database where you could go and you could search for, given the rodent data of a certain age in rodents, what sample sizes do we have in the human cohort, and where can we get to those images? And we also went a step further and loaded in some of the free surfer segmentations and some of the fusion data and some of the metrics generated from some of those data, and then you could kind of do this correspondence type analysis. So we did that in Conti Center 1.0. Conti Center 2.0, we now are taking data from the MRI scanner using Hudocom, and that has the option for the investigators to go directly to bids. We load the data into a flywheel in Mike's lab, or Mike's lab does this. Mithra loads it into a flywheel instance because it's easy for folks to collaborate on that data, get to the images themselves. From there, we export them according to bids and can either share them or use some of the bids to NDA tools to help us in getting that data into the NDA. We're using GitHub for some of the code, but we're not using Data Lab just yet for versioning data sets and so forth, and scripts or scripts applied to those data sets. So that's kind of the state of where we are right now. With respect to using ReproNim tools in that sense. But because the data is in bids, we can, of course, take a next step and maybe provide in the future some versions of the data set or graphs of the information contained in the data set to a Repro Lake instance so that others can find it alongside, Abide, ADHD 200 data, core data set, all these other publicly available data sets that are being used. So,
is this data destined to be part of some official public release in some way, NDA or somewhere else in its requirements?
NDA, yeah. They require us to put the data into the NDA, like most others. And we all have difficulty one time or other using the NDA. So I think, and I'm speaking off the cuff here, but I think that there's probably interest within the Connie Center to make that data available in another way besides the NDA. Maybe a simpler way to get the data. So, things that we have trouble with, our BIS-BAS assessment, the way or our mask assessment, the way we score the variables is different from the canonical version in the NDA. So we have to do a remapping of the scores into a different scale, to push that data in the NDA. And as we all know, that can be error prone. So, sorts of things like that make it difficult for our investigators to feel confident about what the NDA would share from our data set. And so I would guess that we would also like to make it available in other ways, but that remains to be seen as we move forward.
Yeah. One of the other repro mantras, so to speak, in addition to what you've already talked about in terms of standards like bids and cement, markup, annotation, as you've mentioned, and the thought about the issues with data versioning that come with something like data lad. One of the other mantras tends to be trying to get our sort of our workflows containerized into not so much bare metal types of things. Can you say anything about if that's something you've looked at or haven't, or is that a challenge, or just anything?
Well, I might have some additional comments on it, but from my perspective, we have used Docker for some workflows, certainly not all workflows. Of course, we're using HCP or HPC clusters with Slurm job scripts and things like that pull version software, not in containers, but also we're running Singularity containers on those clusters, pulling those Singularity images down from others who have put them together most often. We're still kind of new at creating our own Singularity or Docker images of custom workflows in Mike's lab. I don't know that that's been done yet to make it publicly available, like maybe the PVT or workflow. So we're still new at that. So there's some of that and some, still sort of bash scripts. But I can think the main step we've made in the last number of years recently is, moving to a version control system like GitHub, where people can collaborate on scripts and things like that. So for example, I have a bunch of scripts for pet processing that Mike's lab is using and making contributions to and changes and checking them back in. And that whole process, I think, has gotten better over the years, where we're not opening up a bash script anymore and looking at it and going, okay, I got to interpret each line and go, what are they doing here? What are they doing here? So I think that's gotten a lot better with the help of efforts like ReproNim.
Great. And again, it's a long, slow process. And again, we are so, appreciative of, you talking to us about these and, being amenable to dabbling with these, as we do this. So thank you. Other questions as we come to the end of our hour from the group? If not, I want to thank Mike and David again. Next month, let's see, that's going to be December 2. We have Keith Bush from the University of Arkansas coming to talk to us. He recently published a paper with some of his colleagues, where they used the slowdown of COVID to really attempt to turn their imaging center into a repro center and embrace many of the concepts, during that sort of slowdown. And I have a lovely paper discussing the challenges, of doing that. And so we'll hear in person about their trials and tribulations and successes in becoming more repro center centric, over the past few years. So thank you again, everyone, for joining us, and we'll see you again next month.